Hello, everyone. Good evening in America. Good morning in Europe. I'm Octavio Escudero, President 2024 of the Mexican Gastroenterología Association, and on behalf of the AMG as well as the Mexican Neurogastroenterología and Modalities Association, I would like to thank the ESNM and their executive committee, especially to Dr. Frank Serviv and Dr. Daniel Paul for giving us the opportunity to share with our members this great webinar. This is our second participation, actually, this year. Along with me, Sher is a very good friend of mine, Professor Edoardo Savarino from Università di Padua. As you know, he's a global expert in neurogastroenterology, and we'll be glad to share us this webinar. Welcome aboard, Edoardo. So this is a very special meeting. Thank you to all of you for connecting from both parts of the ocean. And this webinar is part of the webinar series on gut, Microbiota and Clostridioides difficile. And we will be reviewing hot topics on this infection. And we have two great lectures as well as two well-recognized experts. So we'll start with the first lecture. And we have Professor Dr. J. Kuijper from Leiden, the Netherlands. He's the head of the experimental microbiology at the reference laboratory for Clostridium difficile and the Leiden University Medical Center. And he's Professor Emeritus of Experimental Bacteriology, and he will talk about general burden and clinical management of Clostridium or Clostridioides difficile infection.
So, yes, can I share my screen? So can you see me and can you hear me? Is my screen okay?
It is perfect.
Yes? Okay. Thank you, Octavio, for the invitation for this presentation. When I received the presentation, I saw that the time was 3 o'clock. I thought it was 3 p.m., but it is 3 a.m. So it's now middle in the night in the Netherlands. Nevertheless, I'm happy to present some data on the burden and clinical management of Clostridioides difficile infections. You probably all know that the C. diff is an anaerobic bacterium, an anaerobic gram-positive bacterium. It can occur asymptomatically in the intestinal tract of humans and animals, and it can cause hospital-acquired diarrhea, presenting as mild self-limiting diarrhea or as a very severe intestinal disease as a colitis. Increasingly, we do see it in the community in patients who do not have predisposing factors for CDI and are diagnosed just simply because we use multiplex PCRs. The complications of CDI, Clostridioides difficile infections, are the mortality. There's an overall mortality of 10 to 15%. The attributable mortality is strongly dependent on the age and on the infecting strain. That's about 3 to 5% in our country. The most serious complication is the relapse. 20% of all the individuals do suffer from a relapse. And of those who have a relapse, 40% will suffer from a second relapse and 60% from a third relapse. So relapsing disease is an important complication of CDI. And of course, the disease can so severe and affect the colon that there will develop a toxic megacolon with a perforation. So the problem of CDI is, on the one hand, the mortality, on the other hand, the recurrence rate that is so high. So in Europe, we do have about 14,000 deaths each year, but in Europe, it's less. It's 6,000 because we are simply not so large as the US. The most important predisposing factor are the antibiotics. So I will shortly present on the CDI burden, on the guidelines, and on the resistance for the antibiotics that are used to treat CDI. So there's a very interesting publication, that's an overview from a Chinese group published in the Journal of Global Health. and that used a large database from the global burden of the disease, from the resistance map antibiotic use from the CDC, from the FDA, from the adverse event reporting system, and also from the GLASS from the WHO. And they used well-known definitions for CDI, such as the disability or the adjusted life years, and also analyze the age standardized death rates. And of course, what you can see is what you expect, that is there's an association between the occurrence of CDI in certain countries and the use of antibiotics. So the higher use of antibiotics, the higher rate of CDI is observed. And there's an increase of the antibiotic use in the last 30 years. So that is also the reason that there's an increase of the C. diff infections. And as you can expect of those antibiotics, the clindamycin antibiotic is the most frequently used antibiotic that predisposes to CDI. There's an important difference between the age. So this is the use of clindamycin above the age of 67 years. And this is the use of clindamycin below 67 years. So in contrast to all the other antibiotics, if you are younger and use clindamycin, you do have a higher chance to develop CDI. So lincomycin, clindamycin is an important antibiotic. Carbapenems, such as imipenem and meropenem and ertapenem also, cephalosporins too, tetracyclines too, macrolides also, and fluoroquinolones. So if you are, let's say, a treating physician, and want to give an antibiotic treatment that does not predispose for CDI, the cotrimoxazole, so the combination of trimethoprim and sulfamethoxazole, is one of the most safe antibiotics to use. And we do use these antibiotics, especially in epidemic situations when we want to change the antibiotic stewardship. So C. diff is the most increasing pathogen of enteric diseases in the last years. So if you are older than 67 years of age, then you have a higher chance to develop CDI, but also to develop more severe diseases. It's very unfortunate that females are more easily affected than males. That has been observed through all the years, and we do not have a very good reason for that. What is also found recently is that peripartum women who frequently use antibiotics for, let's say, a urinary tract infection or an antibiotic for a sexual cesarea, those peripartum women are also in a high risk group to get severe CDI or relapsing CDI. Mainly, I think, due to the fact that CDI is not recognized. It's lately diagnosed, so the disease already had a severe cause. And if you want to combat CDI, the regions that successfully slow down the increase of CDI, that's based on antibiotic stewardship, infection control measures, and on an effective anti-CDI treatment. So these are the data from Europe. So if you have, let's say, a control group and a case group, and you have a group of patients with recurrent CDI, it's clear that the patients with recurrent CDI do have a longer mean hospital stay of 55 days. That's 2 times longer than the case group or the control group. And also that the costs for individual patients are especially high in the recurrency group. And if you look more carefully to these costs, it is mainly a change of the escalation in the CDI medication, so switching from metronidazole to vancomycin and to fidaxomicin, and also the necessary treatment in an intensive care unit. There has been a patient organization in the US that's changed last year. It's now called the Peggy Lillis Foundation. This is a very active foundation, a very active patient organization. They have also regular webinars and organize all kinds of meetings. And I would recommend this organization for everyone who want to involve patients and who want to ask for advices to patient organizations worldwide. So there's also now a change of the epidemiology of CDI, especially found in Europe. So we see an increase of patients in the community with CDI. And earlier it was mainly the elderly who used antibiotics for, let's say, outpatient treatment. But when we use those multiplex PCRs, that simplifies the diagnostics. We also see young males or young females without any predisposing factors, such as hospital stay or previous antibiotic use, who just develop CDI. And so we have now a tendency in Europe to go to a change of the diagnostic advices for all patients with infectious diarrhea in the community. So we advise now the microbiological laboratories to use those multiplex PCRs and also to report CDI when it has been found. Another interesting new observation is that we do have a metronidazole-resistant strain now that was first found in the UK and subsequently also in Poland and Serbia. That's called the ribotype 955. And that has similar activities as the hypervirulent strain that caused disease worldwide since 2004 and stopped in 2015, 2016. And finally, it's a One Health problem. So especially for the food-producing animals, pigs, you can also find C. diff over there. And that's the same C. diff as is causing this, infections in humans. And people who live at pig farms do indeed have a higher chance to develop CDI. And if you look for the environment of those pig farms, it's not only that you find those strains in the farm itself, but it's also spread, and you can find it in the neighborhood of all those pig farms. So it's not only the 955 strain that has been increasingly recognized. We also have outbreaks of strains that are very much related to the hypervirulent 027 strain, outbreaks in Hungary, outbreaks in Czech Republic due to the ribotype 176. And the ECDC, so the European Center of Disease Control, has collected a number of those strains from different countries, sequenced the strains, and the data has been submitted now as a report to the clinical microbiology and infection. In the Netherlands, we also had an outbreak of one of those strains that was observed in Hungary. And about 2 or 3 years ago, we first noticed an outbreak in Greece due to a ribotype 181. And this type, this 181, is now the most prevalent PCR ribotype in the Eastern European countries. So it has spread very rapidly and affects now mainly the Eastern European countries in hospitalized patients. Here you can see that all those strains are very much related by whole genome sequencing. Of course, you can see that the 027 strains are much more related to each other than the 036 strains, but you can clearly see that this is one cluster. This is one sequence type, the sequence type 1, and that contains different PCR ribotypes. So for those who are viewing this presentation or, let's say, trained in epidemiology, this is a challenge. So if you use the whole genome sequencing, you can't differentiate those different PCR ribotypes. So this is, I think, the first observation that by using whole genome sequencing, you lose discriminatory power. So we advise for the C. diff still to use 2 typing methods. PCR ribotyping, which is very simple, is not so expensive, and whole genome sequencing if you want to look for local clusters and antibiotic-resistant genes. So for the treatments, there are 3 important guidelines, I think, currently. So this is a guideline, of course, from Europe, the European Society of Clinical Microbiology. Sorry, that's my background. But there's also an important guideline from the IDSA and the Society for Healthcare Epidemiology of America, a guideline published in 2021. And there is the guideline from the American Gastroenterology Society for the prevention, diagnosis, and treatment of CDI. So all the three guidelines are from 2021. And they have, in principle, the same recommendations. So here you see a period of initial CDI. This is the first recurrent episode, and this is the second recurrent episode. So the standard of care for is usually now fidaxomicin or vancomycin. We prefer fidaxomicin because it has a smaller spectrum and does not affect the microbiota composition. And vancomycin is still a very good agent to treat the initial episode of CDI. If you do have a high risk of recurrence of CDI, you can also administer a monoclonal antibody against the toxin B to prevent relapses. That's besletoxumab. And if you do not have fidaxomicin or vancomycin, then it's advised to give metronidazole or tapered and pulse dose of vancomycin if you do not have fidaxomicin available and the patient suffers from a recurrence. And for patients with a very severe CDI, vancomycin or fidaxomicin are the preferred options, but we also advise when oral administration is not possible to do a local delivery or to add metronidazole together or with tigecycline. And for the complicated refractory severe CDI, we consider indeed tigecycline and FMT. So there are a few differences. The IDSA, for instance, does not advise fetus microbiota transplantation. However, in a meeting a few months ago, they came back from that decision and they now allow to use fetus microbiota transplantation also for the third or second episode. Also, the tigecycline and FMT for refractory CDI is typically European advice. And American Gastroenterology and the IDSA are not in favor of that. The same is true for the severe CDI when we advise to use tigecycline or metronidazole. That's not taken over by the American Gastroenterology or the Infectious Disease Society. So there are three important antibiotics: metronidazole, vancomycin, and fidaxomicin. Yet disadvantages of metronidazole are large. So it's absorbed and it reaches systemic levels in the blood. The stool concentrations are extremely low because there's an enterohepatic circulation. Also, the inactivation of metronidazole by other bacteria in the stools are high. And it's a typical anti-anaerobic agent, so it reduces all the anaerobic bacteria that are present in the flora, in the intestinal flora, and provides something as we call a colonization resistance. And it does not work on spores of CDI, and it does not inhibit the sporulation. So if you then compare vancomycin and fidaxomicin, you see that fidaxomicin is preferred because of these two options. It does have some activity against the spores, and probably thereby also decreases the relapse rate. And it has a very, let's say, small spectrum, so it prevents a disruption of the gut intestinal microbiota. So for the fetus microbiota transplantation, we do have now in the US 2 commercial options. I hope that Federica will go deeper on those 2 options. I will just mention it briefly. So we do not have in Europe those 2 commercial FDA approved options. We only have in Europe the conventional fetus microbiota transplantation. And that's recently approved according to the, let's say, the European regulation. So we can use it now in national donor fetus bank in Europe. And most of the fetus banks in Europe have been are collaborating strongly together in an organization that's called FMT. And those two options, the RBL and VOS, are extremely expensive, to be honest. And they are now taking over the conventional FMT treatment in the US because the stool banks are not supported by the FDE anymore. So this is a big difference between Europe and the US. Are there new products on the market that are promising? Yes, there are. There is a new live biotherapeutic product now that is that contains 8 bacterial strains, and those 8 bacterial strains have now been used in a phase 2 study and showed indeed beneficial effects to prevent recurrent CDI. And the company, Vedanta, started a worldwide phase 3 study now. And also in the Netherlands, a few of those hospitals are participating in that. So this is the only live biotherapeutic product. There are also new important small molecule antibiotics, Ibezobolstat, also now in a phase 3 study. This is another small molecule, also in phase 2-3 study. This is a new monoclonal antibody, and this is probably also very promising new small molecule. But there are also products that have been halted or withdrawn. So this is the most famous. It has been withdrawn, I think, a couple of months ago. This is an FMT-like product. And the data has been published in a journal called Gastroenterology. But I mean, we don't understand why this product has been withdrawn from the market, whereas the data from the study are very promising. Products that inactivate antibiotics are not a very large success. Those 2 antibiotics are also very active against CDI, but also for unknown reasons withdrawn from the market. And to be honest, vaccines, all the vaccines that have been developed so far failed completely, mainly because it's only composed from toxins and not from cell wall components of C. diff. So do we have to worry about antimicrobial resistance of C. diff to the vancomycin, metronidazole, and fidaxomicin? The short answer is yes. As expected, for all the antibiotics, you can, of course, see some resistance. This is an interesting study. This is a European study from an organization, Combat CDI, that the data were published recently in the Journal of Antimicrobial Resistance. And they look for antimicrobial resistance in humans and in animals for commonly used antibiotics in 4 different European sections, in the eastern part, the northern part, the southern part, and the western part. And for the fidaxomicin, for instance, in the human and animal, also in those 4 different parts of Europe. For metronidazole, that's in blue. For metronidazole, you can see that in the eastern part of Europe, the resistance rate, so the MIC values, is higher than in other European parts, except for the southern part, for instance, because there is also increasing. But in the animals, metronidazole resistance is more frequently observed in the Eastern European countries. If you look for the vancomycin, you can see that vancomycin resistance, that's in orange here, is very rare in both in humans as in animals. For the fidaxomicin, that is in dark brown, you see that also the resistance rates are very low and do not differ significantly in the different parts of Europe. There's also an unexpected finding, and that's the tetracycline-like antibiotic, tigecycline. So tetracyclines are frequently used in the animal industry. So what we expect is much higher prevalence rates of tetracycline-resistant strains in the animals than in the humans. But that's not true. You can see it in the eastern part of Europe. There is very clear that, you can see that, I have to repeat this. So tetracycline use is indeed frequently observed in animals and leads to a higher resistance rates in animals. is the rifampin, the rifampicin antibiotic that is frequently seen in humans, especially in the eastern part of Europe. And that is because in the eastern part of Europe, tuberculosis is frequently found, more frequently in the western part. And rifampicin is an important use for the treatment of tuberculosis. So the resistance mechanism of fidaxomicin is for microbiologists very interesting. So we do have here the genetic data, and this is the RPOB gene. And in the RPOB gene, there are certain areas that are, if they are mutated, they cause resistance to rifamycin, such as rifampicin. But there's one area outside this known area that if there are mutations, it causes a resistance rate to fidaxomicin. However, important is to remember that it's still a mutation in a gene that is already present. It's not a new gene, it's not a plasmid. What we previously found was that it is very rare. It's only a few strains that had an increased MIC value. And if you do have those strains that are less susceptible, is still the question what occurs in the intestinal tract, because the pharmacodynamic data suggests that the levels of fidaxomicin are very high. What is also interesting is that if you do have those mutations encoding for fidaxomicin resistance, the strain is less fit. It survives less well, it produces less toxin, and it produces less spores. So we expect that mutations in these genes will result in fidaxomicin resistance, but will do not lead to large outbreaks. And until so far, there are 6 reports on clinical isolates with reduced susceptibility to fidaxomicin worldwide. I have seen very lately a report from a US hospital in which they described the first outbreak of a fidaxomicin resistant strain. But in that outbreak, there were only 10 or 15 strains involved over a period of 2 to 3 years. So I still wonder if that's an outbreak of just this very rare development of resistance in strains that do not result in spread and causing outbreaks. So when you, as microbiologists, we can download all genetic data from C. diff genomes. And there are currently more than 25,000 genomes available in the public domain. And if we download this data and look for fidaxomicin resistance, we have not found it. And another new mutation in a repressor gene has been described, also resulting in a less susceptible fidaxomicin strain, and also a mutation in an RPOC gene, not an RPOB, but an RPOC gene. But all these observations need confirmation. It's a difficult question for vancomycin, because vancomycin is administered orally and achieves very high concentrations in the intestinal tract. And so if there's a slight increase of the strain from, let's say, 0.5 to 4 or 8 milligram per liter, we still wonder if that's causing clinical failure. And another interesting observation is that all those vancomycin resistant genes can be found in C. diff, the FAN genes. But those genes are incomplete and are not transcribed. So the majority of those vancomycin resistant genes do not cause resistance in the bacteria. So the difficulty is that there is one study published in the Clinical Infectious Disease that reduced vancomycin susceptibility indeed results in less initial cure than vancomycin susceptible strain. However, we need to critically review this manuscript because it's only, so here are all parameters that are important for a 30-day sustained clinical cure, 14 days, or a recurrence rate within the first 30 days after ending the treatment, or the mortality. And only an univariate analysis has been done, no multivariate analysis, because they considered the number of patients that were included in the study too low. And you can see that there are some differences only in the 30 days sustained clinical cure and the 14 days sustained initial cure. And that's not a big difference. It's about 10% difference. So I'm myself not very impressed yet by this study. And I still wonder if what would happen if we do a more larger study with a multivariate analysis. And what is also interesting, this is a study that is published in PLOS Biology. If you do, you can make strains very easily resistant to vancomycin. But those vancomycin resistant strains also have display a reduced thickness. They do not grow very well. They can't sporulate as the parent strain, and the cell morphology is also different. Finally, the metronidazole resistance. So there's a discrepancy between Europe and the US, considering the breakpoints of metronidazole resistance. In Europe, it's much lower than in the US. And there's a complex resistant mechanism of C. diff for metronidazole. We found a plasmid-mediated metronidazole resistance that can be transferred from bacterial species to another bacterial species. But there are also now non-heme-dependent mechanisms and heme-dependent resistance mechanisms. That means that resistance to metronidazole only occurs in a situation in which there's heme, so blood, in the environment. And this is now important because those mutations are mainly in a promoter of the NIMF-B gene. Those mutations do increase the MIC for metronidazole and are also coinciding with mutations in genes that encode resistance for fluoroquinolones. And now we are beginning to understand why that O27 strain that has the mutation and also is frequently fluoroquinolone resistant had the capacity to spread worldwide and cause epidemics. So this heme-dependent metronidazole resistancy is important. It's also observed in the new hypervirulent 955 strains. And it's clearly associated with decreased efficacy of metronidazole in contrast to vancomycin. So my conclusions are that please try to differentiate antibiotic resistance for non-CDI antibiotics and for CDI antibiotics, because the antibiotic resistance for carbapenems and for clindamycin, rifamycin, that facilitate the spread of C. diff between patients and also between countries and world parts. Still, vancomycin and fidaxomicin and FMT or FMT-like products are the cornerstones of treatment, not metronidazole anymore. Fidaxomicin resistance is very rare and not, as we know now, cross-reactive with other antibiotics. Data on vancomycin resistance are very difficult to reproduce. Transfer of important VAN genes has not been observed from, let's say, other bacterial species that are resistant to vancomycin to C. diff. And the fitness costs due to all those mutations for vancomycin and fidaxomicin are important, we think. It remains difficult to assess the clinical relevance for those resistance to vancomycin and fidaxomicin. And partly this is also associated with the lack of standardized testing. So what we found, for instance, is that the heme-dependent resistance rate of metronidazole is strongly dependent on the testing of your strains in the presence of metronidazole, in the presence of heme, because if you test those strains without heme, you can't find those resistant mechanisms. So this is my final slide. I want to thank, of course, the technicians that are doing all the work, our colleagues from the National Institute of Public Health, Daan Notemans and Karuna, and my colleagues from Leiden University. Wietse Klaas is the scientist. He is doing all the research on C. diff. And Geoffrey is a clinical microbiologist and is doing the work for the National Reference Lab. I want to thank you for your attention.
Thank you. And Very nice presentation, very interesting data to show to us, and also thanks for the acknowledgement to the other people involved in your activity. So everyone had his own. I would like to say that there is the possibility to do a question because we have a Q&A chat where you can put your question for our speaker, and so please do that. In the meantime, I think that we should break the ice because I don't see question, or maybe there is one, and I can do that immediately. In many countries in Latin America, there is no fidaxomicin or oral vancomycin. So it is common to use vancomycin for IV use and give it by the oral route, which is quite similar to what happens also in some countries in Europe. The clinical response seems to be good. Is the pharmacology or the bioavailability different with oral tablets versus IV vancomycin given by the oral route? That's a good question.
Yes, thank you for the question. I think this is a very good solution. I mean, also in our hospital, we do not use the vancomycin tablets because they are expensive, and it's more easier to give the liquids, the fluids, prepared from the intravenous solution. So that's exactly what we are doing too in Europe. And is there a difference in the bioavailability, pharmacokinetics? I don't think so. It has not been studied, I have to say. So, but I can't imagine that the vancomycin from intravenous solution differs a lot from the vancomycin in the tablets. So it will probably remain in the intestinal tract and will not be absorbed. Concerning the pharmacokinetics, yes, I mean, there's not so much known about vancomycin and the persistence of vancomycin in the intestinal tract when you stop with the treatment. So you do treat the patients 10 days with vancomycin, high concentrations. And when you stop after those 10 days, if you look then to the disappearance of vancomycin, there's a large individual variation. So in most of the patients, it's completely disappeared after 1 week after stopping of the therapy. But there are some patients who do have longer shedding of vancomycin, also patients who do have it only for 48 or 72 hours. But I don't expect that it will differ dependent on the solution or the tablets.
Thank you for the answer. And there is another one, which is any comments regarding the use of rifaximin to avoid recurrence of C. diff infection?
Yes, also thank you for the question. That's clearly someone who knows the literature and probably has also some experience with the treatment of patients, because rifaximin is used as a tracer antibiotic. So in those patients who have a high chance to relapse CDI or do have a relapse of CDI, commonly the used antibiotics, such as metronidazole, vancomycin, is then supplemented with rifaximin. So rifaximin is a rifamycin, so it's strongly correlating to rifampin, for instance, and it causes very rapidly mutations. And I showed a slide with the presence of rifampicin resistance in the eastern part of Europe where tuberculosis is frequently found, because rifampin is also a part of the tuberculosis treatment. So I think that on an individual basis you can use the rifaximin, but when you want to systematically use this antibiotic in your patient population, then there's a high risk to induce resistance, and thereby the antibiotic cannot be used anymore. So yes, advise it on the individual personalized treatment, but not as a routine part of your treatment for a patient with multiple recurrent episodes of CDI.
Yeah, I agree. That was a very good question, and also the answer was very, very clear. I have another question. Also, Octavio has one.
Yeah, I have a couple of questions. Dr. Kuijper, you mentioned that up to 30% of patients with CDI have known predisposing factors. In these patients, is there any other early life profile that put them in risk later in life, such as use of antibiotics during pregnancy or type of birth delivery or changes in microbiota in early life associated to perhaps use of antibiotics, country of birth, type of diet, or perhaps a microbiota profile developed during the lifetime?
Yes, thank you for the question. I mean, I was hoping to get this question because I think it was one of the most interesting studies that we did in our country. So the study was in more than 15,000 patients that went to a general practitioner and had diarrhea. So when a patient goes with diarrhea to the general practitioner, he already has some form of severe diarrhea because you usually wait a couple of days before you go to the GP. And we did a case control study and we tested everyone for CDI. So irrespective of the physician's request. So that was also our aim, just to look for the, let's say, undiagnosed cases of CDI and to see what happens. And we found many cases. So it was the third most frequently pathogen. So as you can expect, the Campylobacter was number 1, Salmonella was number 2, but the C. diff was the third on the list of enteropathogenic bacteria. And when we did the study, we did not give the results to the general practitioner. But the general practitioner phoned us. And well, can you please inform me about the study data? Because this patient still has diarrhea and it's becoming more severe and I'm thinking to admit him to the hospital. So then we broke the code and decided, okay, it's probably a larger problem than we know and that we are aware of. So that means that in that study, we also gave the results to the general practitioner, and in more than 90% of the cases, he decided to treat the patients. So we do not have any data on, let's say, the spontaneous cause of CDI when it's not treated in a general practitioner. It was a case control study. So for each case, each patient with a C. diff infection, we had 2 controls. And the 2 controls were patients from the general practitioner, the same general practitioner, with diarrhea, but without a clear cause. And there was no difference in those 2 different groups concerning the previous antibiotics and concerning age and concerning other predisposing factors. But we did not look in much detail. So it could be that indeed diet is important and that microbiome composition is important. And that's our subject for further study. But it's very clear that if you're just a general practitioner and you do see a patient with diarrhea and you do not have access to all those questions on microbiome and diet, then it's probably the best to test just for C. diff in a multiplex PCR.
Clear. We have a few more questions from the audience, which probably we may answer. There is one from Jose. Do you suggest any probiotics as coadjuvant treatment?
This is always a topic of discussion in our country and in the guidelines. And we are very strong, and we think that's, and we follow also the Cochrane database analysis. I mean, they were clear there's no place for probiotics. And if there is one of the probiotics that is doing better than the other probiotics, then it's the yeast, the Saccharomyces. So sometimes when a physician does not have the possibilities to treat a patient anymore to prevent recurrent CDI, then he prescribes yeast, the Saccharomyces. But it has not been, let's say, recommended in the guidelines, and it's not frequently used in the western part of Europe. That is probably untrue in the southern part of Europe and in the eastern part of Europe, where probiotics are more popular and more easier also to get.
Yeah, that's true.
Is there any place for Saccharomyces vulgare use in patients with no antibiotic use, perhaps for prophylactic use? Usually,
yes. So that's one of the reasons. And other frequently applied algorithm is when you do treat a patient with a non-CDI antibiotic, and you know that he is in high risk to develop CDI because he already had a previous episode, then they also prescribed the Saccharomyces. But it's personalized medicine.
Yeah, that's true. Not really evidence-based, you mean?
Exactly.
I agree. And maybe we have time just for one additional question. What about nitazoxanidine?
Yes, we do not have any experience with that. So yeah, I can't comment on that. Sorry for that.
That's fine. The same here in Italy, I have to say. And then the other question was related to FMT, but we have now another talk about treatment, so probably there will be time to talk about that. And so I would move with the next speaker. It's a pleasure for me to introduce you, Professor Federica Faccioti, who is a well-known expert in microbiota-based therapy and more in general on basic science regarding microbiota and its effect on human biology. She's an associate professor at the University of Bicocca in Milano, and it's really a pleasure to listen to her about her talk. So please, Federica, it's your turn.
Thanks a lot. Do you hear me?
Yeah, perfectly.
And do you see my
Also,
screen? Perfect. Very good. Thanks a lot. It's really a pleasure to be here. And my talk will be more on the treatments and on the new avenues for the treatment of C. diff. As it was very beautifully described before, the C. diff infection is actually characterized by high degrees of dysbiosis, meaning a loss of diversity and quality of the microbiome. And this is usually, as it was described, accompanied by also a loss of function in general of the microbiome, showing also an increase of an increase of aggressive and pro-inflammatory bacteria and a decrease of beneficial taxa. What is being done to treat the CDI is actually a plethora of mechanisms. They all together try to tackle all the pathogenic mechanism by which the C. diff infect and persist in the host. So the antibiotics that they were largely described are actually taking care of the direct killing of the bug of C. diff, while other types of interventions are more defined in the modulation of the whole community, of the whole ecology of the microbiome, and I will talk about these in detail. There is obviously some new avenues that take into consideration also different stages of the maintenance of the C. diff within the host, like the functional toxins that the C. diff produces. And there are new drugs that have been approved and they are recommended by the guidelines, and also some others that are in pipelines. And as indeed the infection interacts, so C. diff interacts as a pathogen with the immune system of the host, it is obvious that not only the microbiota part is being studied to fight the C. diff infection, but also the immune system functions are being pushed to resist and to combat in a better way. Just in a snapshot, what I was talking in a more broad way, and this also connects with the previous talk. Deals with the bug itself and the infection in the very early stages, but also during the maintenance of the infection status. But the characteristics of the Clostridioides difficile itself, so its capability to be a sporogen, to produce biofilms, render it very persistent within the host, and thus people can become predisposed to reinfection and recurrences. And here comes the new lines of therapeutics like the FMT and the toxins that block the functional, so the antibodies that block the toxins. So it was already very well described. I just would like to add that as much as the USA and Europe are similar and also different, there are also other parts of the world where the treatment of C. diff is really is still different from Europe and US. For instance, like for the use of metronidazole and fidaxomicin, that as we were discussing just a few minutes ago, it's not available everywhere. So I will skip because it was already well described, and I will go straight to this because I want to just make a point. These are the guidelines from the European guidelines, where it's becoming suggested not only to use, of course, fidaxomicin, vancomycin for initial or for high risk of recurrence and so on, but also to try to do combinatorial therapies. So introduce also the antitoxins drugs in those cases that are particularly complicated and may contribute to persistence and recurrence. So the bezlotoxumab is the antitoxin approved drug, but also in the severe and complicated CDI and the refractory severe CDI, here, as it was suggested before, is increasingly being considered the FMT as an option to reshuffle the microbiome communities. And not only bezlotoxumab is being approved, there are some other molecules that target different sites of the toxin, both toxin A and toxin B, and they are most of them still in the preclinical studies. It is also true that antibacterial peptides, as it was mentioned, are being studied again in an attempt to approach the therapeutic armamentarium in a more holistic way. So not only trying to kill the bacterium, so trying to kill the Clostridioides, but also attack the functions, so the toxins, and also potentiate the activities of the host by producing, introducing antibacterial peptides against toxins. And the idea of restoring, so of reshuffling the whole ecological communities of the gut microbiome has a scientific fundament, because at a certain point when the infection becomes severe and refractory, it is very difficult for the ecology, for the microbiome to return to its full functionality, even when some functions are remaining, but the resilience of the microbiome is somehow lost. So one possibility is to completely change the ecology by reintroducing a new community from a healthy donor. And in the last 10 years witnessed really a huge number of proofs that indeed FMT is highly effective in C. diff. when in several studies, the very first from 2013 was a randomized clinical trial in which it was compared FMT with vancomycin, was really showing an impressive result of almost 94% of cure of recurrent CDI. And other types of studies were performed analyzing whether a fresh versus a frozen biomass would make a difference in the efficacy of the FMT, or the source of the FMT, so whether it would better be to use a non-related donor, screened and analyzed very carefully, or to utilize an autologous RCT, an autologous FMT. And another important issue was the dosing of FMT. Since it was not possible to address exactly the composition of the FMT, then it was not possible to dose the function of the treatment. So it was investigated whether a single dose of FMT versus multiple infusions of the microbiome, of the healthy microbiome, would make a difference. And indeed, it was shown that multiple FMTs were more effective because it was promoting the engraftment of the new microbiome. And so the last 10 years really witnessed a huge change in the field of the FMT, because since the publication of the first clinical trial, we moved, as the professor was saying, to new types of products that are more on the commercial, that are actually commercial products. So we are moving from an academical FMT to a product-based FMT, although in the vast majority of the countries, the academic FMT is still the most used type of modification of FMT that is administered to patients. This is mainly due to the regulations. So from this side to the other side of the ocean, regulation is pretty different. So the FDA that approved, as we said before, the commercial products, so the microbiome therapeutics, the Microbiota and the VOVST, started 10 years ago to try to regulate the use of FMT, and it was initially considered, it was put under the umbrella of the investigational new drug. This was wise in a sense that it allowed the people to be treated, so people with C. diff to be treated, and also to start studies, specifically NRCTs, to address specific questions. In this case, it was also understood by these types of RCTs that it was necessary an engraftment to really cure patients, and not only in terms of amount, but also in terms of frequency of the infusions. In Europe, as it was said before, it is a little bit different, and it is different also within the members, the states members of EU. In general, as this year, basically, it was approved the regulation, the directive of the European Parliament that defined FMT as a substance of human origin, aligning it to other substances of human origin like milk, like blood. And this was important because in some countries, FMT is considered goes under the regulation of the transplant of tissues and cells, and this makes it a little bit difficult, as I will reply to the question before, as it is happening, for instance, in Italy with FMT, because it is really considered a transplant, like a transplant of tissue. And this goes under the umbrella of the Center of Transplantation, so the Italian Center of Transplantation. But in order to ameliorate the standardization and the offer of this FMT to patients, within this European directive, new rules have been implemented to distinguish the phases of the manipulation of these products, of these biomasses, in order to allow also the use of commercial, let's say, processed biomasses. So on one side, there will be the FMT, the biomass originated from the donor. So let's say the classical FMT, and on the other end of the story, there will be the product that underwent a certain process. And a topic that was discussed before was the stool banking. Stool banking is really important because this allows for the standardization and also for the availability of material. Because one of the key points of FMT is to guarantee the safety of the product that is infused to the patient, and therefore the process of screening of the donor is very, very precise and time-consuming and also costly. So the possibility to have stool banking allows also to trace the material that is being infused. It is true that the very beginning of the FMT stories started really from an initiative that was that in the end was a stool bank, basically. And it provided, that is the Open Biome initiative that is in USA. It was starting in 2013 in US. And it really provided a huge amount of doses for people with C. diff. Clearly, at a certain point, and you will see where it happened, the drop, it was in 2019 with the outbreak of SARS-CoV-2, of the COVID pandemics. And this was because it started to be several concerns about what it was transferred to people, started to make FMT on one side for sure more safe, and on the other side more complicated to be organized. And indeed, this was also accompanied by the evidence of two patients that actually died after an FMT because they received an FMT that contained drug-resistant E. coli. And also some other patients were actually infused with preparations that contained enteropathogenic E. coli or Shiga-toxin E. coli. So in the end, now the regulation is very, very strict on the types of screenings that are being performed. But this also moved the field towards the preparation, so the studies, the organization of different types of FMTs towards a better standardization, either by FMT-based therapeutics or to prepare knowing which could be the good bugs that can reconstitute the microbiome, so the ecology in C. diff patients, then to generate defined bacterial consortia that could be used instead of the full FMT. And in this sense, the two products that have been approved by the FDA are slightly different, and there are others in pipelines, and I will enter in the details. So the Microbiota is the first one that has been approved. It is very similar to a classical FMT. It is basically based on fecal matter. It is controlled in terms of quantity of the bacteria, of the, let's say, microbiome that is transferred. And it is prepackaged in a single dose microbiota suspensions that cannot be administered by the patients alone, but it has to be administered by an healthcare provider. The trials of the Rebiota, the Punch CD and the phase 2 were interesting because while the first the phase 1 was the primary endpoint was basically safety, was actually working, and it discovered that in this case, one dose was not as efficient as the 2 doses. When it was then moved to the phase 2, where it was compared with the placebo, the 2 doses versus 1 dose, actually it was not, the 2 dose regimen was not statistically different. But it has to be said that the population that was enrolled in the trial was not exactly that is suggested in the guidelines as a good as the population that should receive FMT. So actually the study population was considering people that had either 3 or more CDI infections or more than 2 severe CDI with hospitalization within 12 months, but actually the guidelines consider indication for FMT with people that had episodes, so with the shorter terms. So there was a phase 3 trial in which it was again investigated as an endpoint the absence of CDI diarrhea within 8 weeks of study treatment in different time points, and in this case it actually worked. The number of patients that were enrolled was pretty high, so more than 180 patients against versus the placebo. And here, interestingly, the intervention was 1 dose of Rebiota after standard of care antibiotics with washout. And it worked. It has to be said that, thanks to this trial, then it was approved. There was a difference, but in the placebo group, the recurrence was pretty low as compared to the very first trial, so the trial of ANMN of 2013. So we might say that again, so the study design and the population that were sometimes can be different from the very first studies that showed a very high efficacy of the FMT, or in particular the academic FMT that is being tested again in comparison. One interesting point, all these commercial products do not have particular adverse events, and this is good. And also the interesting thing is that systemically it is being analyzed, the composition of the microbiome after the FMT, and it is shown that restoration is occurring, and upon FMT, the responders increase the frequency and the presence of the good bacteria that usually are present in healthy people. The second approved treatment, so the commercial FMT, is the SER 109, as a little bit different idea. So it is not a biomass that is, or so it's not it is feces derived, but it is not a biomass that is transferred, but it is actually processed. So the stool is processed to enrich in bacterial spores, and in this case are the spores that are encapsulated and are given to the patient. Usually are given 4 oral capsules during 3 days, and as it was said before, the cost is pretty high, so around 17, on average, between $15 to $17 per treatment course. These are data from the insurances. It depends on the insurances. For what concerns the phase, the trials, in this case, the phase 1 was comparing the doses. So it was a trial a dose finding trial. And the primary endpoint was the recurrence up to 8 weeks, and it was really highly successful. Then there was a second, so the phase 2, it was called the EcoSpore because at the beginning this product was called EcoSpore, and unfortunately, the low dose failed. The low dose was not showing any difference with the placebo. So it was performed the phase 3. In this case, again, it was changed a little bit the structure of the study. It was there was a different pre-treatment with vancomycin, with the bowel preparations before the treatment with the SER 109 with 4 capsules. So let's say a pretty high dose. And in this case, it worked, and there were no serious adverse events. And also in this case, we can say that, again, differently from the famous Van Nod paper, there was, again, not a similar rate of recurrence in the placebo and also in the study population that was enrolled. Still, it worked. As it was mentioned before, there were other compounds in the pipeline, including the Finch CP101, that was very interesting because, again, was encapsulated, so it was somehow more handy than treatment by colonoscopy or rectal administration. It was working, but in the end, so the phase 2 trial was highly successful, but in the end, it was not. It was discontinued for no clear reasons. It is instead in a very good progress in the pipeline, the V303, the Vedanta, and again the concept is a little bit different. So while before we were talking about stools-derived biomasses that were either infused as such or manipulated to obtain the spores of Clostridia that could be, but they were always donor-derived. In this case, the concept is to identify good bugs that could fight or could restore the ecology, the good ecology in the host in a rational way. So by knowing what these bugs, these good bugs do. And this Vedanta product was actually derived from very long preclinical studies and knowledge on the functions of Clostridia, not only on the ecology of the microbiome, but also on the functions of the immune system. So this product is composed by a consortium of 8 strains of Clostridiales isolated from a healthy donor and can be administered by pills, so encapsulated. And the phase 2 trial was very, very successful. And there were basically no major adverse events. These are the let's say new therapies that are ongoing, but there are some even newer approaches that are being attempted in also trying to respond to the ever-emerging capability of C. diff to resist to treatments. One interesting approach that is being tried is the fecal virome transplantation. So the ecology in the gut is not composed only by bacteria, but as it was mentioned, by also fungi and viruses. And these viruses can, for a long time, it has been known that can interact in a dynamic way with the communities of the bacteria to regulate their frequency and their functions. So it has been proposed that the transfer of the healthy viruses that are present in healthy donors can be as effective or complementary to the transfer of bacteria or of the treatments with yeasts. And in this case, so they are suggesting the possibility that by transferring the viruses that targets specifically proteobacteria and enhance, sustain the expansion of the good bacteria, this would help in restoring of healthy microbiome. The other possibility is to knowing which are the phages, so the bacteriophages, so the viruses that specifically target, like antibiotics do, the Clostridioides difficile, then one might think to directly infuse cocktails of phages that may directly target the C. diff. And this is interesting because it can also respond also to the ever-growing need of containing antimicrobial resistances. And very last, so I was telling you that there are also attempts to modulate the immune system to render it more fit in fighting the C. diff. And indeed, there are cytokines and anti-cytokine antibodies that target the immune system and facilitate the clearance, and also the small molecules that were cited before that target different activities, both enzymatic and also that activate the immune system, or that target some other metabolic pathways within the host to potentiate the response towards C. diff. So in conclusion, what is currently happening is that by guidelines, antibiotics as Fidaxo and vancomycin are surely the first, the standard of care. There is being suggested and is being more and more used the use of antitoxins in case of recurrent and severe C. diff infections, and for the recurrent and the resistance is being introduced also the concept of FMT. That is not only one type of FMT, but there are different types of FMT, either donor-derived, commercial products, manipulated, spores, defined consortia. And there are some new avenues that probably will help to tackle in a more holistic way this type of very complicated infection. And thanks a lot for your attention and for the invitation.
Thanks to Dr. Federica Faccioti for her comprehensive lecture. It is very interesting to think of CDI treatment not only in terms of killing the bug, but also to modulate the immune system and the microbiota as well, because that opens other therapeutic gates in this exciting era filled with biologic small molecules directed almost against anything, including toxins, as you just mentioned. Well, the first question came from Gilbert Blendon. He asked, he would like to know the experience in Italy with the transplantation of microbiota. And I would complete the question what is the success rate, the percentage of CDI recurrence following FMT, and if there is any difference in terms of success of recurrence according to the way of delivery or number of previous attempts or of eradication with antibiotics.
Okay, so in Italy is, as I told you before, the FMT is under the regulation of organs and cells and tissues, so it's considered a transplant. So only 5 centers currently in Italy have been granted the authorization to perform the FMT. One is actually the center of Professor Savarino that performed a huge number of FMTs. The thing is that it changed a little bit the regulation in terms of severity, but it's actually very good on how to screen the donors, how to maintain the biomasses before the infusions, and the route of delivery. So currently the route of deliveries that are authorized are by colonoscopy, by rectal enema, and also by the nasojejunal route, although it is not used so frequently unless strictly necessary, because there were in the past in some RCTs some adverse events. For the success rate, in general, it ranges from 70 to 80%, depending on the number of doses. It's clear that with the increased number of doses, then you can really reach up to 90%, 95% of the. It also depends on the type of patients that is being treated. These clearly impact quite a lot on the number of infusions that are required, and also on the possibility to engraft very efficiently. And then there was another question. Okay, I don't remember.
Dr. Jorge Suazo from Honduras has a similar question. He asked about efficacy between comparing the delivery mode, if it's better with oral frozen FFT capsules or versus rectal or colonoscopy application.
Allora, this is a very interesting question. So there have been several studies that compared, as I said before, the fresh versus the frozen, the capsules versus the colonoscopy. It is clear that the capsules are more handy for the patient in terms of utilization. It is not extremely I mean, the number of capsules is pretty high currently. So those that are used range from 15 capsules per dose, and it's not pleasant. But it's also true that the colonoscopy as well. On the other hand, the colonoscopy allows a better distribution of the product within the colon. So it has an advantage in this sense. So the distribution is more uniform and more probably allows a better engraftment. On the other hand, the pills are more handy, but also a little bit more complicated in the production and also in the ingestion.
Okay, I think Dr. Kuijper has some comments to add. Your microphone is...
Yes. Thank you very much, Federica, for this presentation on FMT in Italy. We also have FMT in the Netherlands. We have only one center because we are a much smaller country, and our success rate is indeed above the 80%. Because we carefully select the patients who come to our center. So it appears that, let's say, about 20 to 30% of all the patients that request an FMT do not have recurrent CDI. They have another disease. Usually it's inflammatory bowel disease, and then we decide not to treat the patient. So that explains why our success rate is so high, because we have a strong selection for the patient. But what is so interesting is that if you follow up the patients and you ask the patients how it's is going after 2 or 3 months after the FMT, then there's an amount of 30% of the patient that still complains about diarrhea. So they think that the FMT was successful in treating the recurrent CDI, but they still have diarrhea. And all the tests are negative, and the physicians call it an IBS-like syndrome, irritable bowel-like syndrome. I don't understand that clinical disease, so perhaps you have more background on that. And do you see that also after FMT, that patients suffer from this?
So that for what I can imagine, and my opinion is that the success in the engraftment. So when you change your microbiome, you want it as more different from yours to treat the C. diff, but it cannot be too much different in order not to be recognized by the immune system as foreign. So I suppose that this type of responses, so the diarrhea, is probably a response from the immune system that actually recognizes this microbiome as something pretty much too different from its own, and it can induce specific antigen responses in T cells, for instance.
Okay, there's another question by Dr. Kuijper. If bestoloxumab is used frequently in Italy?
Sorry, it was sorry?
If bestoloxumab is used frequently in Italy?
I can answer this question.
Why? Go.
Okay, Edoardo raised his hand.
Yeah. It is not quite common to use it in clinical practice because of the price, which is probably the same issue because it's not used also in the other countries. And as far as I know, also the company who produced bezlotoxumab, Zimplana, they are probably moving to stop the production of the antibody and probably will not be more available in the future. I mean, the additional benefit in terms of reducing the recurrence is small, not so high to say, yes, we can pay 2,000 euro for everyone. And so probably that was the other reason why it did not succeed in clinical practice for its use. I would like also to add something regarding the FMT here in Italy, because indeed the rules we have regarding the fact that if fecal microbiota is considered a tissue and not a drug by our authorities limit a little bit the rules and the possibility to use it in other clinical setting like IBD or IBS. We have to do individual requests for each patient, which is quite challenging in doing them in clinical practice. So we do them quite uncommonly. But for C. diff, for sure, our success rate is about 85%. So it's very, 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 very effective type of therapy for C. diff infection. And also the recurrence is very good. Of course, there are other potential therapies which are more easy to use for sure, and also more available in countries with a lot of population like Italy, UK, or France, which is another possibility, and maybe in the future we will know more. But of course, the cost will be a main issue in order to introduce and have them available in each market. Regarding the other indication, we use them also in some other indication like IBD and DRO, multi-drug resistant organism infection. The results were good, not great like in C. diff, but good. And you're right, Head, when you say that if you select very well the patient, probably the response will be higher than expected.
Okay. Here's another question by Mitsuko Herrera Sato. He asked if patients with IBD and C. difficile infection have a lower recurrence rate if they are already on anticytokines, biologic, or small molecules therapy. Is there any data that is?
I was starting to think.
Yeah.
It's a good question. There are some papers published in literature showing that there is not a major effect regarding the presence of concomitant anti-interleukin antibodies for small molecules. There are some data from the US registry of Jessica Allegretti, who is a well-known expert in FMT, and also in IBD, showing that if you do small molecules or monoclonal antibodies, there is not particularly drawbacks in terms of success rate for FMT. So you can do that quite easily without any problems and probably without major side effects, at least related to the procedure. Then there is the disease, and you have to manage also the disease, of course.
Okay, and I have another question for the panel. One concern is the theoretical risk of acquiring either another infectious or even an autoimmune disease from the donor. And that's why there is this healthy donor profile, including absolute and relative contraindications. And one of these contraindications is previous antibiotic use in the 3 to 6 month previous period. But there is no clear guideline in order to avoid other medications that induce dysbiosis. What other drugs should we avoid before being considered a donor?
I don't know, Heidi, if you want to, or if you want to answer.
Yeah, in the screening, so that in Italy, there is no specific definition on drugs, previous medications, so histories of tumors, familiarity of different types of diseases, yes, but the concomitant or the previous use, for sure, the antibiotics. And I'm not 100% sure about the PPIs, but I don't think so.
Okay,
at the moment, I don't think.
But it's important to say that for becoming donors in Italy, you have to follow the rules of being donors of the liver, of the kidney. So you almost have to be completely off of any disease. So it's difficult that you take drugs if you don't have any disease or any clinical symptoms. So that's the reason why
it's not specified, because it's so difficult to find the good donors in this sense, because the screening is really strict since the very beginning. So the number of potential donors that we get is on average around 6%. We in Milano, we increased a little bit the numbers by starting from the blood donors. So already people that are more prone to donate, but also more, let's say, attentive to their health status. So this is increase the success rate of donor screening up to 20%, more or less, from 6 to 20. But really, it's difficult.
Yeah, that's also true in the Netherlands. About 3% of all the healthy individuals that are screened are finally becoming a donor. So that's very low. But I think it's important to differentiate between donors used for the treatment of patients with multiple recurrent CDI and donors who are used for the treatment of IBD or diseases in which they use immune checkpoint inhibitors or Parkinson's disease or MS. I mean, for we do know that for recurrent CDI, there are not many, let's say, conditions to the donor that influence the response rate. But for IBD, there are some suggestions that superdonors may exist. And for Parkinson's disease, in our study, we did not find any effect unless we looked for the donors. Then there was a tendency that some of the donors had a better performance. So for each disease, you probably have to look for different donor properties.
We actually tried to do this. We are doing a study to find the best match donor recipient in IBD. And we had a matrix basically trying to analyze the response of the immune system and the microbiome compatibility between host and donor. And in some cases, we didn't find a good any match with our bank. That was interesting.
Okay. I think there are no further questions. I don't know if anyone from the panel has another comment, or if not, I would ask to our experts to give us a short take-home message. Dr. Kuijper, take-home message?
No, thank you for this invitation. I found it very exciting just to give this presentation in the middle of the night. And I thank you for all the questions, and happy to answer any further questions by mail.
Okay, Dr. Faccioti, take-home message?
I think we are still at the beginning of understanding some mechanistic issues, but I think that we are on the good route to understand it better and to have more tools in the armamentarium.
Great. Once again, on behalf of AMG and AMNM, I would like to thank the European Society of Neurogastroenterology and Modalities for the invitation. It has been a wonderful session. It is great to be connected in different parts of the world to share medical knowledge. Thank you, Edoardo, for giving us some advices in fecal medical and FMT, in addition to be sharing with me. And thanks everybody online. Greetings from Mexico, and hope you see you soon, guys. Good evening or good morning everywhere, wherever you are.
Good night. Good night for us. It was a pleasure. Thank you, everyone. And
bye. Thank you.
A big hug to all my Mexican friends. Bye-bye.